Mm.
Hello. No? Yes. Should work now. Okay, so good morning again. Uh, Thank you for kind introduction. I'm really, really honored. And thank you for kind invitation, dear Mr. Chairman, dear Secretary, and Dr. Sandeep Pawar. So the topic given to me was organoid modeling organ development, and this is in three dimensions. So I will be touch upon the basis of how organoid development lead to the drug discovery, as well as what are their application in the pharmacology. So the first question I asked myself when the topic was given, why organoids matter. So here I thought of bringing my own experience. So in back in 2021, we had a 2D cell culture based on the patient-derived primary cell culture, and if you see over a period of 7 days how the cell grows. So the cell grow uh, and they form a cluster, and by the time they overgrow, they start forming a spheroid shapes, and this leads to loss of polarity. So this is the problem with the 2D culture. From the same patient, we generated organoids, and if you see when the organoid grow, they really mimic the ductal epithelial phenomenon, which is a uh, fundamental aspect of the pancreatic cancer, and they grow very nicely. They start forming IPMNs, and this mimics the ductal epithelial phenomenon. So clearly from the same patient how the primary 2D cell culture versus 3D cell culture, we clearly see a difference in terms of morphology as well as the physiology. So organoids bridge the gaps between traditional cell culture and animal models by offering human reliable insight at the scale. So if we compare between 2D cells, organoids, and animal models, there is clearly there is a benefits as well as some are the uh, disadvantages. Uh, 2D cell culture is very simple, cost effective, but it usually works with a mono layer. There is no real defined structure, and this also associated with loss of polarity, which is particularly important for the cell subtypes which are associated with secretion, such as basolateral membrane, apical membrane. This is completely lost in 2D cell lines. On the other hand, animal models provide a complex system, but they are associated with species differences. There is always a problem between preclinical to clinical translation, and there is a huge implication of ethical consideration. In the in between of it is organoid, which has a defined structure. One can directly implement human tissues, so the clinical trials can be very very quick. Implementation of organoid, and organoids can be developed such that multiple cell types can be incorporated in one system. So these are co-culture organoids or the IPSC organoid that can have the implication in organogenesis. So, so again, when I thought putting the table where we look at the physiological presentation, as we say, 2D culture has a limited advantage over a animal model. Success rate of the 2D culture is much higher, whereas in animal model it depends on the specialization. The time required for 2D culture is much shorter. Cost is much lower. But genomic stability is much lower because the rate of proliferation is much higher. Also, 2D culture associated with lots of so there is a monocellular content, so it is not heterogeneous. The clinical relevance is much lower because of the point that I talked about. Um, 2D culture can be easily modified by CRISPR-Cas gene editing or other targeted gene editing techniques. High throughput screening can be performed with 2D culture, which is not a possibility of animal models. And the biobanking can be done with a 2D culture, but the animal model cannot have this feasibility at this moment. So if you look at the organoids, they lie really in between it, and they have all the advantages over the 2D culture, and they also mirror some of the limitation from the animal model. So these are the ideal systems. So the organoids are really important in the today's technology for the drug development. So what are organoids? Organoids are the three-dimensional structural models of original tissue and organ, which are derived from the body's own tissue or the stem cell. They are highly comparable to a source of tissue in terms of their tissue structure, cell types, and the function. So organoids, by their origin, can be divided into multiple strategies in terms of organoids can be either tissue derived or the pluripotent stem cell derived. It can be also associated with adult stem cell derived. Pluripotent stem cell derived organoids can also be classified as embryonic stem cell derived organoid or induced pluripotent stem cell organoids. Yeah. So if we look at the discovery landscape of organoid, the major breakthrough event was discovery of iPSC. So Shinya Yamanaka discovered iPSCs, and he came up with a cocktail of four transcription factors which are sufficient to transfer the somatic cell into the stem cell. And these four transcription factors are MIC1, OCT3, OCT4, SOX2, and KLF4. By IPSC later, in year 2009, Professor Hans Clevers from Belgium, Netherlands, he developed a intestinal organoid from the tissue. So that from the intestinal tissue, he developed an intestinal organoid. The media used based on the transcription factor and growth factor. Subsequently, after the discovery from Hans Clever, there are multiple organoids are being generated, which associated with all the tissue-specific organoids. So predominantly media composition of all the tissue-specific organoids are associated or originate from the organoid that are developed by Hans Clever. In the last couple of years, organoid technology take much further leap in terms of organ on chips, bioprinting, co-culture organoids, and assembloids. I will touch upon these all techniques in the later part of the presentation. So what are the essential component to engineer organoid? First is we need a cell source. So it can be tissue from the organ or a biopsy or pluripotent stem cell, either induced or embryonic stem cells. Then we need a cocktail of growth factors, which can be such as Wnt or spondin, then the transcription factors or the hormones such as thyroid hormone, insulin, and so on. Other important aspect that we need for the organoid is the matrix. Matrix is the scaffold that keep the organoid into the three-dimensional space. So matrix are usually matrigels, extracellular matrix component, or the polymeric engineered hydrogels, which are synthetic matrix. Further on, to add to it, with advent of the newer bioprinting, we need some physical cues such as scaffolding to support the extracellular matrix that mimics the basement membrane. Also, we need integrating cues, for example, for bioprinting, organ on chip, multiple organ on chip systems. So these are the 5 essential components we need to engineer organoids. So how organoids are being made? So if we look at the pluripotent stem cells, Maybe I can start with the pointer. If you look at the pluripotent stem cell, it differentiates very quickly based on the composition and forma teratoma, which is which is very complex tissue, but it does not have any differentiation potential. So we need to selectively differentiate into ectoderm, mesoderm, endoderm in order to generate a specific tissue organoid. Uh, so this diagram nicely illustrates if you differentiate without any programming, though there is a spontaneous differentiation which lead to the teratoma formation, which has no real organic meaning. Subsequently, the tissue complexity goes down as we start with the program differentiation. And based on the program differentiation, we can prepare a CNS organoids, heart organoids, colon organoids. You may say any. Yeah. So how organoids are being made? So the source of organoids is usually for the pluripotent stem cell derived organoid is usually either mesenchymal stem cells or mesenchymal fibroblast, which is transdifferentiated to iPSC or embryonic stem cell isolated from the blastocyst. These cells are then processed for the germ layer specification, which lead to the transdifferentiation to either to endoderms, mesoderm, or ectoderms. Yeah. Later, it's further subsequently processed by the enzymatic cocktails to have a morphogenesis, and later there is a maturation. So, based on the program that we use, one can generate organoids for specific organ. Pluripotent stem cell organoid has a huge potential, but it's a complex multi-stage procedure. It is very technically challenging, and uh, though it is possible to use multiple lineage deviation from the single same cells type, but the complexity overweighs the use of the pluripotent stem cell organoids. So, what are the major application of pluripotent stem cell organoid? Uh, First is it can be used for the tissue regeneration. So, by transplanting the organoids, whether the tissue grows and there is a regenerative potential, this can be looked. It has a limited use on the drug development. It is majorly used in terms of the developmental biology. So, how the model developed in terms of how the tissue developed during the embryogenesis. It has a potential in host microbe interaction. Um, Pluripotent stem cell has a huge potential in recuperating patient pathology. So how the patient develop a disease, this can be nicely studied with PSC derived organoids. Also, it has a limited potential in terms of disease modeling. In order to circumvent it, there is a other type of organoid which is called tissue derived organoid. Here, what we do is we collect the tissue either from the biopsy or surgery or from the resection specimen. This biopsy, then we do the fragmentation by the enzymatic digestion. Uh, digested fragment then subjected to enzymatic cocktail, which lead to the organoid formation. These organoids are the usually monocellular organoids. So such as pancreatic organoids are pancreatic ductal cell associated organoids. So this procedure comparatively straightforward. It is highly reproducible, and it is relatively easy to scale. However, because of its nature of single cell, it has a we need to have a culture which need to have co-culture system like a multicellular organoid systems, which we will talk in the next few slides. One of the major disadvantage of tissue derived organoid is lack of microenvironment. So this is the major drawback of drawback organoids at this moment, especially tissue derived organoid. So what are the application of the tissue derived organoid? First and foremost is the recapitulating patient pathology. So this organoid, so for example, if you are taking the organoid from the pancreatic ductal adenocarcinoma tissue to pancreatic tumor tissue, it will grow as a pancreatic tumor tissue. So this can nicely recapitulate patient pathology. It can have huge implication in understanding host microbe interactions. These organoids has a huge potential because of their high throughput nature. It can be used for the drug screening, and it can be easily biobank for the further analysis in drug platforms. Yeah. And uh, this organoids can serve as a model for the tissue growth as well as the homeostasis. So after the development of the organoids from Hans Clever in 2009, there is a ripple effect of epithelial organoid discovery. So if you look at the blue are the organoids which are derived from the normal human derived or tissues. And if you look at the red corner, these are the organoids which are derived from human tumor derived derived tissue. So at this moment, by the end of 2024, we almost cover almost all the tissue where we could generate the organoids for, and we could biobank this. So this has a huge implication and potential in drug discovery. So I just would like to summarize the how organoids are being made. So a patient or a healthy individual either undergo surgery or resection. So the biopsy is digested. Then there is formation of the single cell cluster, which is expanded into the matrigel. So this is the matrix, which leads to the tissue derived derived organoid, which can be differentiated further for the genomic editing and for the functional assay for example, for the drug screening. On the other hand, iPSC organoid from the patient or healthy individual, we isolate somatic cell, predominantly fibroblast or other mesenchymal cells. We perform the reprogramming, which leads to the formation of iPSC cell. Reprogramming is usually done by Yamanaka factors, uh, and that further subsequently we subject to the programmed programmed cocktail in order to generate TS derived organoid. These are differentiate into the functional organoid which has a multicellular potential. So one can generate a whole brain organoid from somatic fibroblast cells in iPSC way. So by this way, but one cannot generate the whole brain organoid from the tissue derived cell. So I would like to bring you to attention to the organoid cell atlas. So if you get the time and if you would like to work on organoid, please visit organoid cell atlas. This is a very useful resource where because of advent of single cell RNA sequencing, uh, this was done in terms of to establish the connection between the primary tissue as well as the organoid. So in the left hand side of the screen, you see a single cell profiling of human primary tissues, and on the right side you see uh, single cell profiling of human organoids. And by the consequence mapping and the lineage mapping, one could understand where the correlation of the primary tissue with respect to organoid. So this whole mapping in human cell atlas give very nice trends in terms of the target identification. So I do request you if you get time to visit this organoid cell atlas. So what are the application of organoid? So application of organoid we talked upon little bit. We touch upon the little bit about disease modeling, precision medicine, regenerative medicine. It has application in toxicology. It has application in drug discovery, host microbe interaction. It has huge potential in gene editing. Uh, Organoids can be functional unit for the understanding omics. It has a huge implication in understanding phylogenetics in terms of how cancer evolve over a period of time. And as we already talked about, it has a potential in developmental study. So over a period of next few minutes, I will just touch upon few of the things uh, in terms of application with the practical examples. So organoids. I just bring in some organoids such as brain organoid, kidney organoid, liver organoid, lung organoid, intestinal organoids, and tumor organoid. These organoids can be used for the disease modeling, and their key features are, for example, apical basal polarity. This is one of the important aspects because of, in terms of if you would like to learn secretion, the polarity is very important aspect. For example, intestinal secretion, the polarity is important aspect which we cannot recapitulate in 2D culture, and this is very hard to uh, get it done. Sorry, in animal models. So organoids which maintain the so for example cryptic villus domains. It can maintain the polarity. It has a mucosal barrier these are the system has a huge potential in terms of understanding secretory potential of intestine. Um, because of their this potential, they can easily mimic the disease such as cystic fibrosis, inflammatory bowel disease. In terms of pancreatic cancer, pancreatic organoid, it can easily mimic either chronic pancreatitis, so fibroinflammatory disorder, or pancreatic cancer. So precision medicine. Currently, what we do in terms of practice is evidence based medicine, and and the approach is one size fits all. But usually, this do not leads to the potent response. So that's why what we need is called individualized, patient dependent individualized therapy, which we call precision therapy. So here I bring in the example of pancreatic cancer patient. So we read the slide from left to right. Um, so we have a patient who has a pancreatic cancer. Um, Either based on the stage of the disease, it get resected, or it has an unresected tumor. Then there is a treatment regimen which is monitored by longitudinal response evaluation, and this patient, based on their status, can be surgically resected or perform a biopsy, or even there is possibility of fluid sampling. This all the samples can be processed to generate patient derived organoids. This organoid can be used for the therapeutic profiling. uh, so which treatment fit best for these organoids? It can be used for the biomarker identification, as well as it can be used for the next generation uh, sequencing or next generation platform. Collectively, all these things can can aid into helping the treatment selected guidance. So, if this patient get a treatment A, but the organoid tell the treatment B tend to have the better response on this patient, so the treatment can be switched to from drug A to drug B can be switched quickly. So that's the application of organoid, and the time required for this is not more than 1.5 months. So this actually saves lot of time, and it can improve the therapeutic response. So cancer is a very complex disease. And I bring in here the example of pancreatic cancer. Pancreatic cancer is very, very complex. It has a very high desmoplasia, which means it has very highly fibrotic environment. It is highly heterogeneous, and it has a very multicellular composition. Because of its high desmoplastic reaction, it is very steep cancer, and it has very poor perfusion. It is also associated with neoangiogenesis, and all these factors contribute to immunosuppression. So ideally, one organoid system cannot mimic all these phenomena. So one can do for high desmoplasma tumoroid encapsulated with fibroblast. These are the co-culture organoid system. One can use the multicellular organoids, and then they fuse together to form a fused pancreatic organoid. One can also do assembloid. In assembloid, what one do is they use the cocktail of the cell, differentially use the cocktail of the cell in order to generate assembloid, which mimics the tumor. Also, in order to address stiffness, one can model the stiffness using synthetic matrigel. For the neoangiogenesis, organ on chip or using the cell line such as Huvex cell line, which forms the endothelial membrane, can be used in order to generate the organoids which are highly vascularized to study neoangiogenesis. And to check what is the implication of immune infiltration, microfluidic co-culture organoid can be implemented. So there is a diverse application for the organoid in order to address the disease in the different aspect. So coming back to pharmacology in terms of toxicology. So organoid based assessment of exposure and environmental risk. So consider a scenario. There is a biobank which consists of hundreds of patients. This patient are tagged because of each patient has a different SNP profiling, and each patient has we generated organoid from each patient. So we have now hundreds of organoids. We plated this organoid in one plate and subjected to different environmental health factor, as well as the uh, risk factors such as microbiome, lifestyle, or physical activity. And one can modulate epidemiological aspect, which we call village in a dish approach, where we can modulate the whole village in a dish, modulate the whole population wide uh, changes changes using the different organoids from the different patients. So coming back to most important aspect, what we are here for today is called drug for the drug discovery. So what is the potential of organoids for the drug discovery? So this is a graph from a company which sells the organoid for the drug discovery platform. And so the front line depicts the use of organoids, and dark line without using organoids. So the company said that our company in last 5 years uh, said that if they use organoid, the delivery time for phase one trial is less than 5 years, uh, and which associated with decrease in the cost in terms of 2.6 billion euros. So it has a huge implication. So how organoids are being used for the drug discovery? So there's a well-established biobank which has a capacity of high throughput screening, which can be also subjected to robust in vitro functional assay. The identified target candidate can be quickly screened by the in vivo model system, where you implant the organoid into the mouse and look at the effect, and then by the collective bioinformatics support, we could validate the target and identify new target. So this has a huge potential in terms of the drug discovery. One of the application of the organoid is phylogenetics. Here I bring a case of head and neck cancer. That these are the two patients. The patient has a biopsy, and then they did a genetic profiling of the tumor, and they found this mutation. So these four mutation are not important at this moment, but just look at the pattern. So they found these four mutation at the time of biopsy, and here they found a two mutation. By the time it went into the passage, earlier passage, there is a complete shift into the mutational burden. And by the time it went into the late passages, there is a complete change in terms of the mutational burden in this organoid. And these organoids actually mimic how the patient develop a cancer in terms of the mutational evaluation of the cancer in the patient. So by spending 1.5 months and doing the phylogenetic study, one can estimate how aggressive the cancer would be. So this has a huge potential in terms of genetic screening for the cancer. So this already being employed for the high diseases such as HCC, so the hepatocellular cancer and pancreatic ductal adenocarcinoma, and this is already in some of the cases already in the clinical practice. So what are the current technological advances with organoid? Organoid, as I talked about, tissue-derived organoid are the monocellular organoid system, but there are currently advances in terms of generating multicellular organoid system. What are the multicellular organoid systems? So with epithelial organoid, incorporation of immune cells, different immune cells, and transdifferentiation of these immune cells. There is incorporation of blood vessel using Huvex cell lines. Then neuronal augmentation using different neuronal and dendritic cells with the brain organoid. One of the important aspects is fibroblasts, because the stroma play a very important role in majority of cancers. So organoids, epithelial organoid with a fibroblast or cancer-associated fibroblast cocktail has a very high potential. Also, in order to mimic the STIP test, one can use extracellular matrix, which are synthetic hydromeric polymers. Also, you might have heard about bioprinting and organ on chips. These are the systems which is highly bioengineered system, very specialized system, where one can bioprint the 3D scaffold in order to mimic the organ architecture and grow organoid in there. It also has the fluidic chambers, which help into the mimicking the flow of the blood, and that help into the understanding the physiology of the disease. These are especially this platform are especially important in order to test the blood-brain barrier systems. Here I bring one of the example of the colon organoid by a chip. So they generated a chip, and by the time and they grow based on the microfluidic principle. They grow organoid in this chip, and as you can see, it nicely formed. It nicely from the crypt and villi, which is mimicking the colon architecture. So this has a huge potential. This is still very specialized and very few labs can do this, but this technique has a huge potential in coming future. So now coming to the current challenges and limitation. So one of the limitation of organoid is accumulation of dead cells, either inside or outside of the lumen, and and as you know that accumulation of dead cell either lead to apoptotic response or the necrotic response. So we need to circumvent this. This can be circumvented by better perfusion techniques such as microfluidic chambers. Then there is a limited control over heterogeneity. So we cannot control how the cells would come upon. Best even if you use the differential ratio, there is a less control on the heterogeneity in organoid systems, especially multicellular organoid system. This can be done by using the screenable or reproducible high throughput models where the ratio plays a role and which can be also used by the scaffolding. And the third and most important aspect is organoids do not recapitulate cellular and physiological complexity. I already talked about this. For that, we need to work on, or we need to have the better technologies such as organ on chip or bioprinting that enable physiological response. How the organoid mimics the physiology. So what I believe the future roadmap would be, we need a better vascularized spot platform. We need a GMP grade matrix and the cell sources so that it can be easily transferable in the clinical practice. We need a multi-organ system in order to transfer physiome on chip in order to understand PKPD modeling as well as we understand the cross organ disease modeling. We need a regenerative medicine. We need to explore more about the regenerative medicine approach. And last but not the least, we need an AI biobank. So, which is a federated organoid repository pair with the machine learning to predict patient response and optimize the treatment. So, what is a future roadmap is the clinical translation, better vascularized organoid system, multi-organ organoid integration, regenerative medicine, and AI-powered organoid biology. With this, I would like to thank you all for your patience, and I am happy to answer any question if any. Thank you.
So, hi, Dr. Ujjwal. It was really an informative talk. You touched upon organoids and organ on chip. So, can you put a light on how we can use these organoids, especially in case of natural products where they are basically a prodrug? They reach the target site after metabolism. How we can use such type of organoids for screening of those molecules?
I, if I understand for the prodrug, so this is very important aspect, and I mean, for that we are not there. I would say we are not there in terms of organoids. So we are at the organ level. We are not at the multi-organ level at this moment. One can one can think of doing about uh, air liquid interface. So there's organoid at one layer and the other organoids. So for example, liver organoid at one phase and the say let's say
Okay,
colon organoid the other phase. But we are very in like infancy in that sense. So we are currently as an organized field, I believe still in the phase of one organ multi cell system. So we are not in the multi-organ system at this moment.
so for that we
But
need.
this will be, I think, this will be achievable with microfluidics and organ on chip in the coming future.
Okay, okay. Thank you.
Okay.
Oh,
Yeah.
yeah. Good morning, sir. It was a good talk. I would like to know how these organoids can be used for immunological estimations.
So that's very nice. So especially in the phylogenetic aspect. So I will predominantly talk about the pancreatic cancer or cancer. So cancer has a mutational burden. So the CAR T therapy, for example, T cell mutation, one can quickly screen over a period of 1 month or 2 months in organoids what is the mutational burden it is generating, and this can be easily transferred into the patient. So This is one in terms of immunotherapy with CAR-T therapy. Other in order to having a augmented immune epithelial cell culture. One can already do co-culture system where how the immune epithelial cell has a crosstalk. This can be easily I would just say easy is a very shallow word. So it can be done with the immune cell as well as epithelial cell and how they crosstalk and what is the distance and what is their spatial correlation. This can be done in organoid system.
Similarly, can be used in diabetes, immune regulation.
It can be used in, I think it can be used in diabetes for sure. Yeah.
Okay.
So for, one need to mimic the disease condition, but it can be used. Yeah, for sure. Yeah.
Right. Thank you.
Yeah.
Thank you very much for the presentation. We have developed in our lab some 3D multicellular model,
Uh-huh.
and we do not use matrigel
Uh-huh.
because we think the diffusion with different drugs is limited. What do you think about
So, this, I believe you use collagen or the extracellular matrix. So, this is very, so when I started working with organoids, matrigel was the gold standard. But the problem with matrigel is it has undefined constitution. So we do not know what growth factor come along with matrigel. So people started worrying about it. Actually, how in the better aspect when we do the biobanking, how the matrigel influence. So the collagen, it's as such as a scaffold has a good potential, but the problem with collagen is uh, collagen tend to form the homodimers when we prepare the matrigel. So the matrix is much more aligned. So cells tend to grow much better in collagen, which is not the case in if you mimic this in human situation. In human, majority of the collagen is heterodimer or heterotrimers, and that is the only one of the demerits of only using collagen. But there is hydrogels which are synthetic and which can be used if you really want to mimic the uh, human ECM. But there is nothing wrong with collagen if you want to do studies with respect to clonal evaluation and so on. So I'm not expecting perfusion, but this is my opinion in terms. Okay. If you not having any question, thank you very much. Thank you. Thank you for your time.
Thank you, Dr. Ujjwal, for providing a beautiful overview of organoids right from their generation to biomedical application and emphasizing on their huge translational potential in the biomedical field. And another thing I want to add on is that you have covered almost entire of my talk that I have to deliver. I have to deliver. So I think I can skip that. So now I would request Professor Prakash B. Diwan, a prominent Indian pharmacologist and academic leader with over 4 decades of experience in pharmaceutical sciences, to felicitate Dr. Ujjwal. And Dr. Diwan is the founder director of NIPER Hyderabad. Welcome, Chair.
and it is certificate that we all Thank you.
So our next speaker is Dr. Rohit Dev, scientist, regulatory testing, pharma and medical devices, people for the ethical treatment of animals, PETA, India and Delhi. And Dr. Rohit Dev holds a PhD in pharmacology from Rajiv Gandhi University of Health Sciences. Oh, 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 sorry. So I call upon Dr. Rohit.
Good morning, everyone. So, myself, I'm Dr. Rudbish. I'm working as a scientist at Regulatory Toxicology Department, Data India. The science department, Regulatory Toxicology, is affiliated to Beta Science Consortium International, a big umbrella in Germany and UK. So, all the scientists which are working on the PETA is affiliated to Beta Science Consortium Germany. So, that is our main science big department. So, today I want to share some information regarding the non-animal methods. Before going to that, I want you to just think about the statement. It's not just a statement, it's a reality agreed by all the scientists and all the renowned scientific agencies that about 95% of drugs which are showed promising results in preclinical animal studies are failing clinical trials. So this figures 95%. It's a very critical figure if you consider about the amount of time a drug takes to develop, move from lab banks to the market, 10, 15, 20 years. And the amount of money involves billions of dollars. So this figure is just like a signal um, that, it's signal about it's a high time that we should decide whether we want to turn back and want to invest our money, resources, energy in refining all the traditional models, or we want to move ahead, move forward, adopt these non-animal methods, which are very human relevant because very close to human physiology, and they reduce the time which take by the drug development and testing, and they give you a real scenario, the data, the real pictures, what can happen with the drugs in the human physiology, and save time, save money. So today's talk is about information regarding the NAMs, what type of NAM models available commercially in the market, what regulatory toxicology or regulators globally thinking about the NAMs, what type of advancement or guideline modification recently happens globally regarding non-animal methods, what PETA is doing in India, and what type of progress we have achieved in India. And at the last, we will talk about challenges and opportunities. These challenges and opportunities are very important when we consider about developing these system in academic, or whether we want to integrate in the regulatory system, or we want these non-animal methods adopted by industry. These are especially in perspectives of Indian scenario. There are a lot of challenges, and now there are a lot of positive things are happening by the government, regulatory agency, and scientific agency in promoting these non-animal methods. So I will start with the animal testing, as we have already discussed in yesterday's talk also. So there is about 92% of the drugs fails in the clinical trials. Some of the investigations also say that about 80 to 89% of clinical research is irreproducible. This is also related to academic research also. This is a huge challenge for reproducibility of data. Uh, some studies shows that about only 10% of promising studies transfer to the clinical, trials. And this failure rate is more than 95% when we consider some specific disease or complications like Alzheimer disease, cancer, HIV vaccine development, stroke, and sepsis. So, considering these challenges, limitations, the data scientists have developed one document known as Research Modernization Deal. So the main idea behind this research modernization now document is about giving an overview about the limitation and challenges with animal test models. What are the factors responsible for these failure or challenges, limitations associated with the animal models? What are the regulatory perspectives regarding these animal testing or animal research, including the viewpoint insight of all the renowned scientific agencies? Then, what are the different biomedical research sections like cardiovascular, cancer, and different others, where what type of animal models are available? What are the challenges with these animal models, and what are the alternative models are available? And at what stage they are, either they are validated, or what type of research outcomes we have received on all those NAMs which are under pipelines or under discussion with the regulatory body. So main point or vision while developing this document is the replacement of animal methods with non-animal methods in area where animal models have already shown to be poor surrogates of humans. So What we are talking or what we are expecting from the academic researchers or regulators or the industry is that it is very difficult to remove or replace the whole animal model in the case where the in vitro models or trained models are not available, like Alzheimer's. There are challenges. People are doing their best, but still there is a challenge about developing these alternative models in these disease conditions. But where there is alternative models are available, commercially available, you can consider, you can work out and try to shift from the animal model to the non-animal models. Critically reviewing additional area of research to define whether the animal use can be phased out. So there is one, if you search with the other countries, there are databases where you can find out how much animal is used for the particular research. So we are in communications with the regulatory agency and we are trying to develop such database in India where we know that like one research which is focused on the cardiovascular complications, this much of animals is used this year. What is the output? So any other new investigator who want to use that model, he knows what challenges the previous scientists have faced. Is this model is right selections or not, or whether there is an alternative which can give a better result. So these type of databases can give you a good ideas about number of animal use, what are the output of these animal models. So that can help you in the selection of that models also. Redirecting funds from experiments on animals to non-animal research. So recently when we communicated with the scientific agencies like DBT and BIRAC, now you have seen that there is a special funding opportunity for the development of in vitro models for diseases efficacy. We are still communicating with the scientific agency like DBT, BIRAC, DST to release separate funds for the development of non-animal models because funding is also a challenge in this areas. Introducing a ethical harm benefit analysis system into policies government governing the use of animal in biomedical research. This is related to our databases where we, if you will submit, this is communications going with CCSEA. Formerly, it is known as CPCA. So we are working with them to develop such database where we know that how much animals are issued, what type of research you have done, and what is the output of that research. Is it benefit to use that animal amount or number of animals? There is a possibility where you can reduce, apply the principle of 3R. Either you can refine it, replace it, or reduce it. So the possibility there, and what is the output of your research proposal, like research, whether it is significant or not. And education and training the scientists non-animal. This is also important aspect of PTA initiatives. We organize a lot of because the most important challenges is that hands-on experience which we have faced while we communicating with the industry also. These are new models commercially available, but most of the technology is outside of India. You have to, and the technical specialty or the experts are not still available, but we are communicating with the industry and we are working with those test providers to travel to India, organize workshops, hands-on training workshop. We are working with the National Institute of Biologics, NIB, and in 2026 we are organizing a hands-on workshop on in vitro models, testing model, including in vitro methods for detection of pyrogenicity in the biologicals. So we are also doing it with the regulatory or quality government quality control labs. So, with starting with the drug development, when we discuss about different sections, I will also discuss about what we are doing, what changes have happened. So, slide by slide, you also note what changes are happening outside, like globally, and what changes is happening in India. So, when we talk about drug development, this is one of our projects, which is equine-based therapeutic products. Equine-based anti-serum antivenoms, they have a risk of mild allergic reactions to serum sickness, and there are life-threatening uh, challenges there. Risk of transmitting infections or diseases from host to human, which is very important when we already have gone through the pandemic. So this is very challenging and important. And batch-to-batch variability and short plasma half-life. This short plasma half-life is very important because we can't have a stock. If you gone through the recent article from the government scientific agency about the rabies. This is one of the challenges of availability of the vaccines or antivenoms to the core areas of India, and that is because we can't store or file them for a longer period of time. So the solution is to move to the recombinant technology. So we are working with DBT Virak also to promote the recombinant technologies for the development of anti-sera. Those are companies who are working with the equine serum. Now, most of them have shifted from equine serums to the recombinant monoclonal antibodies. They are still developing some of the company, but that is for veterinary use. Second, we are working with CCSA on equine guidelines. So these equine guidelines, if you go to the CCA guidelines, these are 2001 guidelines. So we are working with CCA. So recently we have a meeting with DCGI. We had a meeting with the DBT Virat new division called Regulatory Affairs and Policy Advocacy. Dr. Sonia heading that division, new division. And we also worked with the Ministry of Health that we need to review this document because from 2001 to 2024-25, the blood collection, other standing operating procedures are totally changed. New advancement has happened. So the CCS agreed. They constituted new committee which are working on this modification of this equine guidelines. And we are also involved some equine experts from the industries to give their inputs to refine these documents. We develop collaborations with the companies to develop monoclonal antibodies. We have a collaborations with the AppCam to develop monoclonal antibodies. We have collaborations with AppCalis for the development of monoclonal antibodies. We are working with US FDA, the previous equine-based monoclonal antibodies. We are transforming them to the recombinant version. We are also, apart from this regulatory advocacy, we are also into research and development. So we have developed this recombinant monoclonal antibody for diphtheria enterotoxins. This project is initiated from WHO because one of our team member is also a part of technical team of WHO. So we are all the regulatory technical teams. We are there where our presence are there. So we share the recommendations time to time. So this request come from the UK HSR Health Research Organizations, UK and the WHO that they want to transform from the equine-based diphtheria toxin to the recombinant toxins. And apart from that, we have also developed some recombinant antibodies. Now, we'll discuss about the non-animal methods. So, first we have the three-dimensional human cell-derived models, which constituted reconstructed human tissue models. We have spheroid. Dr. Ujjwal already discussed about the organoids. We have bioprintings. We have precision cut organ slices. Then we have traditional cell-based assays, which we use a lot in our research. We have in silico models. And then we have a combination models where we have organ on chip. Sometimes we use organoids on chip. And then we also combine some other system with the chip systems for the um, screening of the drugs. So I will discuss about those models which are already available in the market. So these are companies, leading companies, Matex, Skins. These are the models which are already validated. These are the few examples. They have a list of products which are already validated, which are already integrated in OECD test guidelines and used by the industry and other researchers. So this is one of the model. This is Epiderm, which is used to evaluate topically applied compounds. It is already integrated in OECD test guidelines for skin irritation, skin corrosion, phototoxicity, skin hydrations, dermal drug delivery, dermogenicity, epidermal differentiations. So it's not about, these are test models are also used in India. Companies are using this. So this is the simple schematic illustrations how these 3D tissue models been developed. So these are other tissue model, 3D tissue models like epialveolar for inhalation toxicity. Then epioculus, which is used for either ocular toxicity, corrosion, drug delivery. Most of the OC test guidelines, if you see, most of the test models are generally tested with the chemicals, agrochemical pesticides. Very less you find the test model which is tested with the drugs, pharmaceuticals. Most of the information is with chemicals, agrochemical pesticides, but they are now also tested and for the pharmaceutical drugs also. We have Epiderm. So there is, if you go to the MedTech website and you go with the OECD test guidelines, they have the huge list of these models and the test guidelines numbers and their applications. Even though they have the details where you can find out the which model is at what phase of approval. We have spheroids. They are simple cluster. They are different from organoids because they don't require scaffolding to for 3D cultures, and they can't cell assemble or regeneration. So this is a simple method of developing spheroids. Conventionals have some limitations. No, now the scientists have been going for the advanced cell spheroids, known as smart spheroids, and they can be used for delivering the therapeutic molecules in the body. Uh, I will not take this along for organized. Dr. Ujjwal already discussed well for the organized, but now organized are very much popular and accepted by the regulatory body. US FDA granted the first approval of the use of organoids and organ-on-chip technology in a new drug development research. US National Institute of Health announced it's awarding 87 millions in contract over 3 years to launch a standardized organoid modeling center. And in October 25, this is the company which for this organized system got first FDA IND approval for the anti-cancer combination therapy, which is tested on the organoid model. So a lot of regulatory integrations, acceptance from the regulatory bodies happening for the NAMs. This is just an example of a company which is having this model, you know. So these are the advantages with these models, you know. And there is 3D bioprinting, very much utilized, which can be used as target cell selections, lead identifications, preclinical studies, clinical trials. They are combined with organoid cell cultures to form as organ-specific 3D cultures, and then you can, you know, as per your requirement, you can form that environment of a particular organ. 3D bioprinting machines, I think MIT University, IIT Delhi. Selling is a company with which we are working on this bioprinting companies, and they have a huge engagement with the regulators and the industry, where they are providing these type of companies, and the academic institutes where these type of machines has already installed, and they are developing these in vitro models. So these are the other areas where these type of models can be used. Uh, Even though in monitoring drug safety, these type of models can be used. In silico models, we have worked with our partners and we have developed this OECD QSR toolbox, which is free available and which is also under the regulatory guidelines. So these tools can help you in real identifications of toxicology aspects of your chemical entity. So we do partnership with the regulators and industries to develop in vitro models also, whether it's in organoids or any other or in silico. This is US FDA accepted this PBPK simulation for the identifications or screening of the drug molecule. And this also help in reproposing the old drugs with the new drugs. So, but nowadays the regulatory acceptance of in silico is quite challenging unless it is coupled with the in vitro models. So whenever you see applications for new drug approvals, they do in silico regulations, but it should be coupled with the in vitro uh, correlations or corrections or checking of the studies for regulatory submissions. Most of the companies have their own benchmark applications. Those are free on the website, will give you some ideas, but they are not as specific as their own patented benchmark systems. Then we have macrophysical systems, organ chips. Now we have organ chip for each and every models. Dr. Ujjwal is asking about multi-organ systems. So there is one university in US who has have recently developed the multi-organ systems in a one chip, and they are working on the validating on it. It's quite challenging, but things are moving forward. So this is a typical setup of organ on chip system, microfluidic setup. Maybe one layer or two layer. It depends upon your requirements. So I want to discuss about this organ on chip system. This is a liver on chip which got US acceptance for their iStent pilot programs. So US FDA have the iStent programs. These programs, they help the innovators in developing their in vitro models. We can also use the precision cut tissue slice systems for the identification, screening. These are the area: drug discovery and development, disease modeling, biomarker discovery, cancer immunotherapy research, gene therapy, and personalized medicine, where the precision tissue slicing systems can be used. Now, this is important sections where I want to discuss about the global regulatory shift towards. So first, I will start with US FDA. I'm highlighting here some specific recent changes. But if you go to the US FDA or European Commissions and type about non-animal test method, you will see a lot of changes and different sections, like in vaccines, in other drugs and the medical devices also. There are a lot of changes. There are a lot of expenses. Um, I'm just highlighting some recent changes which is related to our projects in India. So in 2017, they released this toxicology roadmap, and 2021 they advanced a new alternative methodology of FDA. They released it. So this is important, which is happened in 2023. This is US FDA Modernization Act, where they have accepted all the in vitro models um, for the preclinical investigations of new drugs. So as FDA Modernization Act happened, after that, the Ministry of Health have also revised the new drug and clinical trial rule, and now they have amended it. And if you check the new drug and clinical trial rule amendment of 2023, they have accepted all the alternative method, including these organized everything, silico computational models there. They have not removed the animal, but they have accepted that those scientists or the industries can use these models also for the investigation of new drugs. In 2025, FDA announces plans to phase out animal testing requirements for monoclonal antibodies. We have also working with the US FDA for this in US, beta US teams. In India, we are also working. So here we have proposed one in vitro in silico model, couple model for screening of monoclonal antibodies, where we can investigate their tissue binding, their receptor binding, no, it's fine. So, and immunogenicity. So, we have discussed these plans with the industry and DBT to execute these type of studies, and most probably we are in talk with NIT to, because it's specifically working on the biologicals. So their labs can execute these type of studies, and we want to validate it and share it with regulatory bodies so that it will be integrated in our guidelines. FDA advances drug development innovations by establishing iStent. I already discussed with you the iStent program for US FDA. FDA medical. So this medical device development tools. This is the program where I'm also working one of my projects, which is on pyrogen testing of medical devices. And this medical devices testing with the rabbit. You know about pyrogen testing, right? How it does with the rabbit, and what is the challenges with the pyrogen testing with the rabbits, because it is this system which is considered as the gold standard has not been validated. Lot of variability is there, when you inject a drug and then uh, insert the temperature, and then you want to see what is the fluctuation temperature in 3 6 9 in numbers of rabbits. So there is a lot of variability. So we want to use in vitro methods like monocyte activation test, which consider both um, endotoxin pyrogen and non-endotoxin pyrogens, because rabbit and mat is the only method which have a wide spectrum of detections. Other than that, we have bacterial endotoxin test in the Indian pharmacopoeia, which we are also communicating to include this, integrate this mat in Indian pharmacopoeia. But that BETs only detect the endotoxin pyrogen, not non-endotoxin pyrogen, which is very important and significant. Recombinant factor C is there, recombinant cascade reagent there. Again, it is only for endotoxin pyrogen, not for non-endotoxin pyrogen. Non-endotoxin pyrogens covers each and everything, whether it's virus, fungi, or the material which is came out from your medical device or the container during the leaching process. So the sensitivity of these monocyte activation tests is very high, and the spectra as well. So it is very important for medical devices also. So we are in communication. We have conducted some studies of monocyte activation tests with medical devices, and we have communicated these data with the FDA also. So in 2025, the agenda focus on implementing novel methodology. So these are all the step by step, year by year, what the regulatory bodies are doing for the advancement of these promotion of non-animal methods. So there are different reports they publish about non-animal methods, alternative methods, validation, qualifications. So if you go to their website, you will see that comparatively here they have years ahead in terms of non-animal method development, qualifications, validations, testings, market availability of these models. They also do the collaborative work. They have performed in vitro cell models for cardiac complications, which is now integrated in ICH guidelines. They do across cutting FDA applied research liver microbiological systems. They do, 3D model of human lung airways for evaluating permeability, inhalation toxicity. They also do collaborations with the stakeholder to validate those models. So regarding the European commissions, there are different reports being generated year by year where they have projected their commitment towards the phasing out the animal methods with the non-animal method. This is more important that um, European Pharmacopoeia has already, removed their normal toxicity tests. They have substituted in vitro methods for vaccine development, replace the histamine sensitivity tests, and this eliminating. Now the European Pharmacopoeia Commission decided they completely removed the rabbit pyrogen test from their European Pharmacopoeia with alternative in vitro validated tests. So this will be enforced by 2025 end. So they have also released the guidelines regarding that, do the collaboration studies regarding the development and advancement of in vitro models, whether it's in silico or whether it's in vitro models. They have recently released non-animal method in science and regulations where they project their commitment towards. So these are the different stakeholder collaborations studies for the advancement of different type of in vitro models. This is OECD test guideline. This is just a small part. You can see the models, the OECD test guideline numbers, and the regulatory authorities which use. So there you go and you will find that there is a lot of in vitro models which are validated, integrated, because we have a separate division for OECD integrations. We have a team which help the industries to validate their models and integrate in the OECD integration test programs. In India, in 2022, we have granted a fund to a company called Biomedical Technology in Bangalore. They are working on the 3D tissue models for eye, vagina, and one more area, I think lungs. Their models already validated at the end of validation, so they want our help for OECD integration. So Department of Science and Technologies, if I'm not wrong, Dr. Hati is the representative for India for OECD coordinator. So we are working with them for the OECD integration. But because for your validate method to be integrated in the OECD program, you have to do a lot of tests with different chemicals. So what we are doing in the advancement of animal tests in India. So we work with a regulator, policymaker, scientists, industry for the advancement on animal methods. I'm leading projects in India for pharmaceutical medical devices. We have teams which also working on chemical, agrochemical, pesticides. These are different type of projects. Some of the projects we run globally, which include food safety, animal-free meat, cultured meat is now also gaining popularity. So we are working on animal-free culture media. You mostly used animal-based cell culture media, but a lot of research and is going on for the alternative as animal-free or animal component-free culture media. We have, target animal pet safety test. Recently, Indian Pharmacopoeia have removed this TABC requirement from 13 veterinary vaccine monographs. We work with so these are the different projects we run. We do technical recommendation, proposal communication with regulatory and scientific agency. We do R&D collaborations. We are now have a developer collaborations with National Institute of Biologicals, where we are validating the monocyte activation test for the pyrogen testing of biologicals. Recently we had communications with the leading biopharma companies in India to discuss the challenges and the opportunities for adoptions of monocyte activation test. In that meeting, we have almost all the MAT test provider globally: MAT Research, MAT Biotechs, CTL MAT, Immunospots, Lonza, Merck. Everyone was there. So it is a test provider, stakeholder, and regulators joint meeting. And next, we are organizing next year in-person workshops for monocyte activation tests. We are working with academic scientists also for the establishment of these in vitro models in their labs. We do help in the developing strategies, roadmaps, and as a contact point with regulators and funding agency, how we can help them in establishing and these in vitro models and during in their studies, either PhDs, postdoc studies, or we also help them in framing up proposals to DBT Virats. Recently, we have a meeting with DBT Virats on fundings for in vitro models. We provide research grant also, like I've already mentioned that we provide a research grant of near about 10 lakhs to Biodimension Technology to validate that, and then we are helping them for OECD integrations. Last year, we sent about 7 to 10 Indian scientists, regulators, and industries to Indian Institute of In Vitro Sciences (IIVS) US for hands-on training experience on in vitro test methods. We provide hands-on training opportunities. We are in communication with Director of NIT in 2026. That proposal is accepted. Now it is communicated to Ministry of Health for approval. In 2026, we are organizing this hands-on training, which we have provided in IVVS. Those IVS scientists, experts are coming to India for providing hands-on experience, and the host institute will be National Institute of Biologics. So it contains all the cell-based in silico, and we have also proposed to include the monocyte activation test as a hands-on there. So here I want to discuss about the small highlights of pyrogen testing, which I'm working on. So these pyrogens are fever-causing agents, especially in parentals, vaccines, biologicals. We have these complications. These are two types: endotoxin pyrogens and non-endotoxin pyrogens. For endotoxin, non-endotoxin, we have rapid pyrogen test, which is already mentioned in Indian Pharmacopoeia as a gold standard. But now Indian Pharmacopoeia commissions have removed this RPT in majority of monographs and replaced it bacteria endotoxin test. So we have also, pushed this issue from a long time. Now we are working to remove this remaining RPT and BET and include not only to integrate the alternative methods in pyrogen testing method. That's why we have developed a collaboration to do a validation of these alternatives in India, because this is the regulatory requirement what we have informed from regulatory bodies, that we have to do the validation studies of these models, which is already validated, a lot of literature, but it is validated out of India. So we have to do validation in India in Indian scenario. So we are working on that. So for endotoxin, we have BET, which is now in Indian Pharmacopoeia. We have now recombinant factor C, recombinant factor R. This BET is based on the blood of this horseshoe crab. So this is ethically is having a limitations. It's detect only endotoxin pyrogen. So when we go for the biologics testing, we need to test the safety endpoints considering the non-endotoxin pyrogens. So for this, we have monocyte activation tests, which have a wide spectrum of detections. We have two type of technologies in there. We have a PBMC cell-based. Now, industry are now more interested in PBMC-based math. We have monomax also, but have some little bit limitations. But PBMC is easy. Either you can use a fresh bird, or either you can use a cryo preserved cell lines. Uh, some of the Indian biopharma companies are using this PBMC-based cell lines with the med test provider, because they are supplying their products to European company, and this is must they have to do according to their guidelines. And we are uh, working with Indian IPC expert working group committee, which now on 25th I am invited to present this validation proposal. So that because IPC we invited IPC to be a collaboration in this validation proposal along with the NITs. So these are all the development which has happened for the starting from rapid pyrogen test. Now European Pharmacopoeia is on the deciding that they are completely removing the RBT with alternative test method. US FDA test guideline for industry also mentioned the importance of monocyte activation test. They have also included recombinant factor C, but it is on the, they have given the provision that the industry can use the alternative models provided them it is well validated. Brazilian Pharmacopoeia also accepted the new journal chapter on monocyte activation test. Indian Pharmacopoeia has a monocyte activation test journal chapter. In 2018, we are working with IPC to modify it because it's very short as compared to European Pharmacopoeia. It doesn't have that much detail about the validation part. So we are working with IPC on that. So recently we are working with the IPC expert working group where we have raised this issue about pyrogen testing. This is a small highlight. We are working with IPC on this pyrogen test for years so that the RBT is removed. Now BT, now we are in that BT is not good. It's better to give another in vitro model integration so that give an options to the manufacturer. They can, who want to go for the in vitro, they can go. We recently had a strategy meeting with all the Mac test providers. We have a consistent meeting where we have DBT, Virat, we have IPC, we have all the industries. Now we are into the final phase of deciding the product specific validations for which we have selected monoclonal antibodies. We are doing the validations with biologicals first, and then we are then do it for vaccines and other monographs. And in 2026, we are also planning to organize the MAC workshops. What recent changes happens? These are the some highlights, but if you go to the Indian Pharmacopoeia, you will see in veterinary vaccines, even though the human vaccines, there are a lot of changes. In potency testing, they have included CHO cell lines as a alternative test methods. Abnormal toxicity test completely removed. Target animal batch safety test removed 13 vaccines. New drug clinical trials accepted the alternatives. Biosimilar. We work with the biosimilar manufacturers in India. They authorize us to talk with the regulators on behalf of them. We prepared a position paper because the previous this biosimilar market authorization in India requires in vivo toxicity testing for market authorizations, but no regulatory bodies in the world require that. So we communicated with that. We sit with industry, take their views. We sit with DBT, Virat, and Cityscope. We understand their challenges, and finally, recently, we got the new revised guideline of biosimilars where they have removed the requirement of in vivo animal toxicity case by case. But they have a separate sections. You can go to the website available on Cityscope also. That there is importance to implement the principles of 3 R's. Animal models have their limitations. They accepted in there, highlighted there. And we have in vitro models and advanced analytical techniques which can be used in alternatives to animal models in terms of biosimilar. So rapid pyrogen test has been replaced with BET. Now we are working on alternative test integrations in pyrogen testing for biologics first. We are also working on replacement or integration of animal free cell culture media in Indian Pharmacopoeia. If you go to the general chapter of in vitro in vivo techniques for vaccine development, now the addition will come in 2026. They have accepted our recommendations. They included the sections of animal free cell culture media in the general chapters. So in vitro test has been added in the IP vaccine monograph. CHO cell line test is been removed. Rabies anti-sera monographs, which if you go, there is a test called monocyte mouse neutralization test, MNT. So now we have some work with the industry. We have submitted the validation data. Now IPC has to select whether to choose RFIT or ELISA as the alternative method. Most probably ELISA will come as an alternative model in the rapid anti-sera monographs. Mass neutralization test is no longer used by any test labs. We have DBT funding recently announced for in vitro model. Pharmacy Council of India course swim test we have communicated. Now, what important thing which I want to discuss with you also, we are working with academic scientists on one thing is that and the students' perspective is also important that, but because I'm also from pharmacy background, I did my PhD from University of Auckland, New Zealand, spanned research a lot of time on research. As I did research project in AIMS, Kansas universities. I'm a DBT postdoc fellow, completed my postdoc from planning. So I worked a lot with these in vitro test models and nanotechnology. Ocular is my field, ocular drug delivery technologies. We want to introduce one module on alternative testing method in pharmacy syllabus, B.Pharm final year and M.Pharm, because this is the time, because what we have studied this, like the curriculum, how much it is revised or how much it is very close to the industries. If you go to the websites, in the company's website, their research R&D, you will see how much they are working with alternatives testing models. Regulatory changes, you are seeing some of the things I highlighted here, but if you go to the website, you will see how much a significant shift is going on globally in India about the in vitro test models. So it is high time that we can introduce a general chapter modules of alternative non-animal models in pharmacy education. So we are developing an expert working group models. I had a chance to meet with the faculties of Jamia Hamdard, where I given a talk on alternatives. MIT University, Chitkara University. Soon I'm going to NIPER also to discuss with the pharmacology department. We want a concern from all the top 10 universities that what pharmacologists are think about these proposals, what are the challenges, if there is any modifications they want, if anything they want, then they will communicate, prepare a draft and communicate with Indian Pharmacy Councils. Because this is very important, our syllabus should be very close to the industries. There are the companies like Akasta Health Science, Biodimension Technologies, BioOnco-C, uh, these are the companies which are working a lot with the organoid models, 3D tissue models. There are a lot of training programs they organize. I request the faculties also to communicate these things with the students of masters, that whenever they have these time points where they have this some training phase also in BPharm, you have to do some training, for 3 months or 5 months or 6 months. They should go to their labs, visit their lab, even though the academic lab, like Dr. Ujjwal is working on organoid, don't do a detailed research. Just see how this system works, how they develop, have some idea. After this, also, if you go to the higher studies in abroad, also, uh, they do all type of testing there. You can integrate these type of testing, but at least you have some basic ideas, so well prepared for industry and for the research. So these are the opportunities. So this is very important. Last and very important, which I have. Uh, After a long discussion with academic, scientists, industry, and regulators, there is a lot of requirement of awareness. Technology details is still unclear under in industry or academic scientists about the technologies. Hands-on training, crucial issue. Industry, academia, even though regulators also, we have arranged some hands-on training with regulators also. That's why, because they want to understand, they have to understand, because ultimately they get the applications for the approval and the validation study results. Supply chain system, because see, most of the tests are available outside. You have to, so there are some issues with the supplies of cells, when we tried for our monocyte activation test, but there are some area where we can work with the regulator and the ministry to make these challenges with the customs and supply relatively flexible for those where their intent is clearly defined, time period is clearly defined, where it is used, how it is used, and the time duration. Because still supply is it takes time. Regulatory compliance, we are working with the regulators also for making, promoting the use of non-animal method. Funding is also an issue. We are working with the regulators, scientific agency like DVT Virat to ICMR also to release a separate fundings for development of in vitro models, or support a consortiums or group, either academia or industry group, academia, industry or regulator group, to validate or develop a project or wide study. We are recently we have discussed one proposal with DPT about developing a centralized facility, because in academia also, if I think, if I talk about the academia, there are three levels of institutions. Those having good amount of funds, those are having so, and those who don't have funds and relative less facilities. So these type of centralized facilities for non-animal methods will help the students, faculties, and industry where they can, get the details about the technology. They can have a hands-on training of that technologies, and if the industry or anyone who want to test their compounds or molecules, they can do that there on validation, and it will be in the government quality control lab. So they have built a good confidence among the users also, and in the regulators, that every step you do is well reviewed under regulatory. Research and development is a lot of requirement there. Collaborations we do with the industry and academic scientists. If you have some ideas where you think that these models can replace, regulatory test models, animal-based model, then we usually support this. We do a lot of collaborations with the industry in the development and advancement. This is what I'm talking about, centralized management facility. This is very important. And the educations, yes, we have one proposal to, introduce one modules on alternatives in the syllabus of pharma. So we do a non-animal based session, different conferences. We provide hands-on training at IVS. We provide travel grants for that. We also give instruments, but it is very, the application is very competitive. IVVS, we have donated some of the instruments for inhalation toxicity. But if you have some projects which really have a transformation potentials to change the animal model, because we are working in regulatory toxicology, so basic research we can't consider unless it has the potentials for transformations and regulatory modifications or replacement of animal models. We provide grants for in vitro models development, advancement. We do research collaborations, do different type of application collaborations. But whatever we work, research we do, publication we do. If you go to the Beta Science Consortium International website, we always do in collaboration with regulator and industry. So we always do whatever we do with regulators, so that every step is reviewed. We know that we are going according to the regulatory guidelines, and every step is well reviewed and well defined. Yeah, thank you. If you have any question,
Dr. Rohit,
yeah.
Dr. Rohit, we are all highly privileged to have a speaker like you today over here. I really appreciate your work with research and refinement of the testing methods. Myself, Sunil Kodi, I am from Serum Institute of India. I work in Quality Control Department. So it was a really pleasure to have your talk here. I mean, it was closely connected. And I would like to know the future scenario of math test. I mean, would it be a real replacement to the BET test, or it is? I mean, there are a lot of developments in BET right now with RFC methods coming on. So, how closely you think the non-significance of non-endocrine pyrogens is related to biologicals, and how are you looking at replacement of BET, or it is an additional math test?
So recently, sir, because like we had a meeting with the industry, Serum Institute of India is there. So after that, we also had a second session is about a meeting with regulators, scientists, and scientific agencies. There we have a BIRAC representative, we have a representative from IPC. So that discussion is totally focused on FP. We have got some suggestions that actually we want to start with rituximab as a test molecule because it's very simple. We don't want to start validation study with the complex molecule or complex vaccine. You know that it will end with nothing. So we want to start because this is the first study. We want to see how it performs, what type of challenges we face, and our motto is that we want to develop a protocol, a unified protocol which industries can use or which can be included in a general chapter as a general protocol for that particular drug. For monocyte activation test, we got a requirement from regulatory bodies that we need to perform the validation. That is from CITESCO and IPC. We developed a collaborations within National Institute of Biologicals for that validations. That validation proposal is already submitted in IPC, is already reviewed. On coming 25th, they invited me on expert working group to present that proposal. We also invited BIRAC and IPC to do a collaboration with that, but they want us to focus more on the monographs where we have a rapid pyrogen test. We don't want to go into small molecule because there is no benefit of going into that. But they have give us a chance to explore more with blood related products, some simple vaccines and other molecular activities. So they asked me a list. Second thing about monocyte activation test is that we are not asking IPC this time or regulators to replace it completely with BET. BET will be there. Our ask is that you just include monocyte activation test as an option to the manufacturer who want to go with MAC. Slowly, when the things, because you also know, sir, that it's new and it's a complex technology. Once the confidence develop among the regulators and industry, it will automatically, once, not today, but tomorrow, it has to become. So we are starting from now so that we get all the validation data, because after that, we have to do a comparative studies with different test kits also. So we are starting with one, but as we have discussed with Virat also, that if we plan for a wide validation study where we consider three different molecular entities, blood group, blood related, one vaccines, one molecular antibody, then we have to select a different type of test providers kits. Then we can, design a big. For that, we need, because we can support. We have funds. We can support testing of validations for one test kit providers with one molecules. Like for the wide spectrum, because Virat also agreed that validation is very important part from the new technologies. They said that if you have such type of proposal, we can discuss it how we can do a collaboration with the industry. You already doing a lot of work on that establishment in India, and we need such companies who are having the expertise on that, and we need their inputs also during the validation study. It's complex. I agreed with that. This technology is not very easy. It takes a time. That's why we are also inaugurating hands-on workshops at NIB for the industry, so that they can do something, do these hands-on and see the confidence develop step by step. So now, next step, we are going to contact with IPSCO. We are sharing the proposal, because IPC can give inputs on the validations. But what type of result regulator expecting from MAC validations? That thing is, I think that CDSCO is the right regulatory body who can give us some guidance or ideas, like what type of results they are expecting. They want us to do a comparative study. They want us to do a comparative study between the test kits, or comparative studies among the RPT is required or not. What type of parameters they see is fine for product specific validations, because it's very clear in the European pharmacopoeia, but Indian pharmacopoeia, their specifications is not clear. So we are also working with IPC to, include more information on match and assessment. So these two things are going parallelly. Sir.
I know it's a challenge.
Yeah.
Very actually very lucid way of when get the most of things. And it's so difficult to probably what you're in for. I think the researcher, I think the workshop for researchers are a much
Yeah.
should be seen as an opportunity. Now, my question here is when you're looking at even some of the process, what is the percentage for argument you are trying to use? So there are a lot of mistakes as a user that concern. How much the input of artificial intelligence particular for this type of test? Because even if the artificial intelligence has even taken, they take a home and a home and the reference place. So whether such type of things can be done without doing much work as far as in the
So with the um, artificial intelligence, there are different models which are accepted by or recommended by the regulatory authorities. But when you do the such studies and go with only the in silico or artificial intelligence data, regulator will not convince with them. They'll give you a big picture, hypothetical, but to cross check the outcomes, you have to conduct the in vitro model. But there are some companies which recently I met with them. I just forgot the name of that company. That company developed a platform which is a coupled model of in silico and in vitro model, where you just put your inputs of your drug, and it will, generate all the possibilities, like which area it can be used. Either you can, it's good for cardiac complications, liver complications. It is a good candidate for cancer. or it. So they have that platforms, and they are also working in India with some of the companies for anti-venoms development. Uh, I think they are working with the Rout Serum, and they have also developed collaboration with Indian Institute of Science for anti-venom development. And that company has a very beautiful, very fascinating platform, and that is also validated internationally. So it is not like a R&D based model or something like that. So these type of couple models are coming, and the regulators also encouraging this type of coupled model. We have also done in silico screening of our recombinant monoclonal antibodies for that map, for immunogenicity. Uh, in the regulatory IPS guidelines, they have a rabbit model for immunogenicity, but everyone knows that rabbit is not a good for immunogenicity. So we conduct the initial study of immunogenicity with in silico model, um, and then we have to confirm those outcomes with the in vitro model. So those in drug discovery companies who are working with the in silico model for drug discovery, it's fine. They have their own benchmark platforms, very effective, regulatory accepted. But after that, you have to conduct the in vitro study to confirm that.
Dr. Ravi, for highlight sensitizing of non-regulatory. Like I cited as a competitor, KPM in policy of development and drug safety to promote non-business, and this is really commendable. And it is also in line with the CR principle of research. So thank you so much. Now I request Dr. P. Sandra Rao to manage this policy. So thank you, Dr. Dr. Sundari, please join. Thank you. Data needed and the center level server. Very good all of you. Transport in the round.
for this very prestigious conference and the pre-conference workshop, and thank you very much for inviting me. And this talk is basically based on sprouting ethics in science, allium cepa as a non-animal model for genotoxicity and cytotoxicity studies. Before that, I will show some pictures of my beautiful country, the Sri Lanka. So Sri Lanka is a country with a natural beauty. So I'm inviting all of you to visit Sri Lanka for our future conferences and join with us. And these are some of photographs of my universities in a campus which consists 8 faculties, and I'm from Faculty of Health Sciences. I will start from basic. So I hope that all of you know what is toxicity. The toxicity refers to the degree to which a substance can cause harm to living organisms. So it may be a chemical, drug, or a natural compound, or whatever thing. Then the toxicology studies are scientific tests used to understand how harmful a substance is and what organ it affects and what doses and under what conditions. These studies help determine whether a substance is safe not only for humans, whether it is safe for an animal as well as for the environment. And when we get different toxicology studies, depend on the duration, we can classify those as acute toxicity, then subacute, subchronic, and chronic. And if it's like a 24-hour test, we can call it as acute toxicity. And if it's like 28 days, then it's subacute. And if it's 3 months, that means the 90 days, it's a subchronic. And if it's more than 90 days, that is chronic toxicity test. And these acute toxicity studies help determine lethal dose and immediate hazard. Then the subacute toxicity is in the subacute toxicity studies that we are giving the test compound repeatedly. So the animal will exposure repeatedly for that test compound over weeks. And these studies are important to identify organ-specific toxicity and safe exposure limits. Then when come to the subchronic toxicity studies, here also there is a repeated exposure, and this study is also important to identify organ-specific toxicity and the safe exposure limit. Then this chronic toxicity, those are important to identify the toxicity that's caused by prolonged use. And when we evaluate toxicity, there are different types like the genotoxicity or the mutagenicity. So by using these tests, we can test whether a substance damage DNA and cause mutation. And sometimes some substances, they can affect on immune system and can make immunotoxicity. Likewise, you can see there are different types of toxicity like the reproductive and development toxicity, neurotoxicity, ecotoxicity, carcinogenicity, like that. And why these toxicity studies are important, they are essential for drug development and also the chemical safety assessment and the food and cosmetic regulation, and also to check the environmental hazard. So they are important for environmental protection and also for the workplace safety. And when come to the topic how toxicity is measured, we can use one of the toxicity evaluation method, and we can observe the no observed adverse effect level. And if we see that this no observed adverse effect level is present for the higher dose, then we can determine that it has no detectable harm. And also another parameter, the lowest observed adverse effect levels. At there, we detect the lowest dose where harm is detected, and the LD5, the lethal dose, that is the dose that kills 50% of test animals, And also we can assess the safety margin and the exposure risk assessment. And when you see the literature, you can see there are different types of toxicity studies, and some are in vivo models which use animals, and there are in vitro models, then in silico models like the computational and AI models, and also there are ecotoxicological models, alternatives, or a new approach methodologies, and the ex vivo models. Likewise, you can see there are lot of methods. Some are in vivo and some are in vitro. And when we get these models, and if we see the advantages and the disadvantages of these methods, we can see there are advantages as well as the disadvantages or a limitation in all of these models. None of the models are give 100% accuracy results, because all these have some kind of limitation. So here I'm not going to read one by one. However, in the slides you can see when we get this in vivo model, it may be a rodent model or a zebrafish or a rabbit. Okay. So one of the advantages of these models are we can use the whole organism. So we can see the whole organism response. And it is important to understand the metabolism and the pharmacokinetic of that organism. And this is good for chronic studies. However, when we are talking about the animal studies, we know there are a lot of ethical concerns and animal welfare issues. In other than to that, when we do animal studies, and if it's a chronic one, it's expensive and also time consuming. In other than to that, we can see the species differences that contribute to reduce the human predictivity. Likewise, there are so many limitations. And then when we get the in vitro model, like the cell lines, the primary cells, and also then the organoids. Okay, so there are some advantages like that we can do this under the controlled environment, and those are some alternatives. However, when we come to the limitation, there are lack of systemic interactions and also limited metabolism. Likewise, we can show limitation. And also here you can see some advantages and disadvantages of other methods also, like the in silico method and also organs on chip. Okay, likewise there are certain methods, and as I mentioned in earlier, we cannot expect that none of these methods give 100% accurate one. They are accurate, but the thing is there are certain limitations. Okay, so we can show limitation for any of methods. However, when we are talking these in vivo studies, because in early time, we can see when we look the literature, in the most of the old research papers, they have used animals. But with the development of technology nowadays, luckily we can see there are so many alternative techniques like organoids, then the organ on chip, and also the 3D bioprinting. Okay, likewise. And when we come to this 3R principle, because when we are talking about animal welfare and the ethical issues regarding use of laboratory animals, okay, so it is very, very important to adhere to this 3R principle, which is known as the replacement, reduction, and the refinement. The replacement is that use non-animal method whenever possible. Then the reduction is when we are doing animal studies, that we need to use a minimum number of animals need for valid result. Because if we use over number, then in animal side it's not good, because in the most of toxicity studies, finally we are euthanize the animals to get the organ. If we use an unnecessary numbers, then it will harm to animals. In other than to that, it's a very costly one, and also we are consuming our time, effort, and everything. So it is not good. On the other hand, if we not get enough number of animals, then it will not give a scientific validity for our results. Because of that, when we are doing animal studies, it is very, very important to do a sample calculation and get the correct minimum number of animals to give a scientific validity. Then the refinement is when we are doing our research, we should minimize the pain, and also we need to improve welfare of animals that we are using. So these three are the 3R principle: the replacement and the reduction and the refinement. So when come to these alternative techniques, which I mentioned in earlier for the toxicity studies, we can use these in vitro methods as an alternative to animal testing in toxicity studies. But when I come to the Sri Lankan situation, okay, so luckily in India I can see most of alternative techniques are applied. But when talking about the Sri Lankan situation, although there are globally so many alternative techniques are available, like the spheroid and the 3D bioprinting, like that, we have very restriction or the limitations due to these financial issues. So we need to do our research with the minimum available facilities. So always through some of our societies, like the Sri Lanka Association for Laboratory Animal Science and the Society for Alternative to Animal Testing, what we are doing, we are always trying to aware our students and the young scientists about the very low cost alternative techniques that can apply to any laboratory but give a validated, reproducible results. In other than to this financial constraints or the lack of facilities, there are some other challenges like the technical and the expertise gap, and the lack of validative alternatives and infrastructure, and the reporting and the reproducibility concerns, like that. So as I mentioned in earlier, always we are trying to introduce a certain methods, the alternative techniques to our students, which they can apply those methods in any lab and can do their research work. So we identify this Allium-Sepa model also as a very good alternative technique that we can use to assess the cytogenotoxicity and the genotoxicity. So it can be used as a choice of simpler or the cheaper organisms or a model to do this cytogenotoxicity or a genotoxicity. So from my today's speech, I like to highlight some important points regarding this Allium-Sepa model. So when come to the cytogenicity or the genotoxicity, the cytogenicity refers specifically to the ability of a substance to cause structural or the numerical changes in chromosomes. And these cytogenetic damage include the chromosomal breaks, then gaps, deletions, then translocations, and the micronuclei formation. Then genotoxicity refers to the ability of a substance to damage genetic material inside a cell. And this damage can lead to mutations, then the chromosomal breakage, and also the cancer development and the reproductive defects. And when come to this very simple method that Allium-Sepa or the onion bioassay, this regarded as one of the best systems for evaluating cytogenetic alterations of xenobiotics. And when we are talking about applications of Allium-Sepa test, this model is used in a variety of scientific and environmental contexts, including environmental monitoring. So we can use this method to assess the wastewater. It evaluates toxicity of treated and untreated wastewater sample to determine environmental safety, and also the pesticide screening Okay, so it's important to test agricultural chemicals and residues for genotoxic potential before environmental release. And also, this method can use to identify cytotoxic effects from contaminations by heavy metals such as the lead, mercury, cadmium, etc. And also, this allium SEPA test is important to toxicological studies, to screening of chemicals, pesticides, then the food additives, dyes, and pharmaceuticals for cytotoxic and genotoxic effects. And also, when we are working with natural product regions, the natural products extracts, this is important to evaluate the safeness of these plant extracts. And also, this allium SEPA is a good educational and the research tool. It's commonly used in biology and the toxicology laboratories for demonstrating the mitotic stages and the chromosomal aberration. And also, it's an alternative to animal testing. So, this one is recognized as a non-animal test model suitable for primary toxicity screening before in vivo studies. And why this anion or the allium SEPA is important and why we are taking that, because it has the rapid mitosis. The root tip cells divide frequently and providing abundant data points for chromosomal analysis. And also they have large chromosomes. So as they have these large, easily identified chromosomes, we can easily observe them under the microscope and can identify different stages of that. And also it's a very cost effective method. It is inexpensive to cultivate and maintain compared to animal based assays. And ethically wise also, it's a very good alternative technique. It eliminates animal testing while maintaining scientific rigor and the reproducibility. And when come to the assay parameters, there are three critical endpoints: for the cytotoxicity, genotoxicity, and the mutagenicity. So under the cytotoxicity, it measures cell death and inhibitions of growth in root meristematic tissues, and under the genotoxicity, it detects chromosomal aberrations and breaks in DNA induced by test chemicals. And under the mutagenicity, it identifies the permanent alterations in genetic material that may be heritable. Then when we get the xenobiotic exposure, they can make macroscopic effects as well as the microscopic effects. Under the macroscopic effects, that we can see some abnormalities in the root shape and the growth rate and also the hardness of the root and the color. Then under the microscopic effects, that we can identify the permanent cell death like the apoptosis and the necrosis condition, and also we can identify the frequencies of mitosis and also abnormal cell divisions with aberrations, micronucleus formation. And each of these measures indicates different effects of test substances and inhibition of rooting, and the appearance of stunted roots indicates the retardations of growth and cytotoxicity. Always when we do these tests, we are using the negative control, so we can compare this root growth for our negative control. And when I get the overall method of this allium SEPA assay, first we need to make the bulb, the preparation of bulb. Then we need to initiate roots. Then we can exposure those roots to the test substances that we need to test. And we can get the macroscopic and the microscopic measurement. So let's see one by one. The first one is the bulb preparation and the selection. Under that, we need to choose the healthy allium SEPA bulbs with uniform size to ensure the consistency across replicates. This uniformity minimizes the biological variation in the cellular response. All of you know that when we do assays, we should minimize bias, and we always should validate it and also think about the reproducibility of our studies. So always we need to get some precautions to avoid bias. So that is why here we need to get uniform size allium cepa. So then it minimizes the biological variation. Then the preparation methods. Then we need to carefully remove dry brown the outer scales without damaging the basal plate where roots primordial are located. Okay, so when we get the allium cepa, that we can see this brown color basal plate. So we need to carefully remove it, and this preserves the meristematic tissue integrity essential for uniform root emergence. Here you can see this meristematic region that I am talking, and here you can see the root cap. We need to carefully remove the basal part. Then the root initiation phase. Once we remove that, we need to introduce these allium cepa bulbs to the growth medium. Normally we are using distilled water. So we can take the allium cepa bulb and we can put it into the the water container by slightly touching with the water vapor. Or we can get the wet tissue and we can keep the allium cepa bulb on the wet tissue for this rooting. Then the optimal root length, this allows roots to grow to approximately 1-2 centimeter in length over 2-3 days. And this length ensures sufficient activity dividing cells in the meristematic zone for analysis. Then for the quality control, we need to monitor the root emergence daily and discard bulbs showing abnormal growth patterns, fungal contamination, or delay root development to maintain experimental trigger. Because if we are not get the healthy root, then we don't know whether if something happened from the allium cepa or if something happened from our test material. So always we should be careful to get very healthy roots when we use these for our experiment. And here you can see a photograph that how we can root in these allium cepa bulbs, and here you can see the rooted one. Then exposure to test substance. So then what we can do once we select these allium cepa roots, we can get the measurement and then we can introduce them into our test material. So we can use the concentration gradient of our test material. In other than to that, we can use some negative controls and the positive control. Negative control may be a water, and as a positive control, we can get any region that we already known as a genotoxicity or the cytogenicity causing agent. Then exposure duration, maintain exposure for 24 to 72 hours depending on study object. And acute toxicity typically requires 24 to 48 hours, while chronic effects need 72 hours or longer. Then control group, always maintain control bulbs in distilled water under identical environmental conditions to establish baseline mitotic activity and the cellular morphology. So likewise, we can expose these roots to our test substances, and we need to take at least triplicate from each concentration. Then the cytotoxicity assessment. Here we can measure the root length as well as the shoot length as some macroscopic parameters. And here measure root length before and immediately after exposure using a calibrated ruler or the digital caliper, and record measurements to the nearest millimeter for accuracy. And then we can calculate the inhibition of these growth of these roots by using this formula. In other than to that, these are the parameters that we can measure. That we can measure the root length and also the hardness of the root tips and the color differences. And in other than to that, we can measure the growth retardation of the shoot also. Then this root growth inhibition we can use as a sensitive toxicity marker, and the root elongation provides a rapid, cost-effective toxicity indicator. and reduced root length directly correlate with the substance toxicity levels. And this parameter is particularly valuable in environmental screening, offering the reproducible results across diverse test substances and concentration. And when come to the dose-response relationship in root growth, there is a control baseline, and also when we are selecting the concentrations, that we can select the low concentration, medium concentration, and the high concentration, and we can check the toxicity for these concentrations. And then we can plot a graph of root length and these concentrations, and then we can measure this IC50 value, and we can get an idea about those test substances. In other than to that, all those are macroscopic features, the root length and the shoot length and everything. In other than to that, to check the cytogenicity and the genotoxicity, that we can use these tips to make microscopic slides. That there what we can do. First, we can take these roots and we can do the root tip excision. That means the cut 1-2 millimeter of section from root tip containing the meristematic zone. At there, we need to use sharp razor blade to ensure the clean cuts and preserve cellular architecture. Then the fixation process. For the fixation process, that we can use ethanol glacial acetic mixture, normally 3:1 ethanol to glacial acetic acid mixture for 10 minutes at 60 degrees Celsius, and this preserves the chromosomal structure, and also it helps to prevent autolysis. Then the hydrolysis treatment, for that we can use 1NHCl, and we can heat our sample 60 degrees for 5 minutes, and this softens tissues and facilitate the same penetration without damaging to chromosomes. Then we can do the staining and the mounting process. In here we are using a stain that called the acetocarmine. So we can put that acetocarmine into our root tips, and then we can put cover slip, and we can gently squash it to spread cells into the single layer for observation. Here you can see how we can do it. So first we need to get tip, then we can fix it, then we can hydrolyze it, then we can do staining, and finally we can mount it on a slide. Then when come to the microscopic analysis, that we can count the cells. Here examine at least 1000 cells per treatment group is required. Then we can calculate the mitotic index. That's so we need to count the cells in all mitotic phases. Then in other than to that, we can observe the abbreviations, and we can record all chromosomal abbreviations like the bridges, fragments, and etc. Okay, so as I mentioned in the previous slide, it is important to get minimally 1000 cells, and it's ensure the statistical power and the reproducibility, and also it is we should consider about this exposure window. It may be 24 to 72 hours, depend on the requirement, and these hours of chemical treatment depending on the toxicity assessment goal. Then the root length target, the centimeters of root growth optimal for meristematic cells analysis. In other than to that, we should maintain the consistent environmental condition throughout all experimental stages to minimize the confounding variables and ensure the reproducible results across replicates and studies. Then in here you can see some of the phases of these mitotic divisions. These are healthy cell stages. So when calculating this mitotic index, we can count all cells in this mitotic phase, and this mitotic index quantifies the proportion of actively dividing cells within a population, serving as a primary indicator of cytotoxic effect. And here you can see the formula that we can use to calculate it. And decrease mitotic index signals the cytotoxicity and the cellular stress response. So if we see the high mitotic index, that means our test extract or the test substance has some safe indication. And when come to the chromosomal abbreviation, the chromosomal abbreviations represent the structural or numerical changes to genetic material. And this presence indicate potential genotoxic or mutagenic effect from the test substance, warranting further investigation and risk assessment. And here you can see some of the chromosomal abnormalities. So we can, when there are abnormalities, we can see the chromosomal thickness and the bridges and lagging, and also the micronuclei, C metaphase arrest. Likewise, there are atlas. So when the students are doing this kind of research, first of all, they need to follow these atlas and they should familiarize with these stages. And this is the data sheet that we can use to record the data. So we need to count these different phases of the mitosis, as well as we can count different the alterations, gene alterations, the chromosomal alteration in those particular slides. And interpreting aberrations frequency, the low frequency, that means the normal background aberrations and the minimal genotoxic risk indicated. And when come to the high frequency, the clear evidence of DNA damage, the substance requires the regulatory concern and further testing. And when come to the micronucleus test, the micronuclei are small extranuclear DNA containing bodies formed by chromosomal fragments and the whole of chromosomes that lag during cell division. And their presence definitely indicate the DNA damage or the spindle aperture dysfunction, making this test a gold standard for genotoxicity assessment in regulatory framework. And here you can see some of the mechanisms for this micronucleus formation. So it is due to the chromosomal breakage or the mitotic dysfunction, and also the nuclear envelope formation, and the high frequency of this establish the genotoxic potential. And integrating multiple parameters for complete risk assessment. The comprehensive toxicity evaluation requires the parallel analysis of mitotic index, then the chromosomal aberration, and also the root growth inhibition, and we can use the shoot growth inhibition also, and micronucleus frequency. And this multi-parameter approach provides the robust evidence for mechanisms of action, regulatory classification, and safe exposure thresholds in environmental and occupational settings. And when come to the quality assurance and the best practice, the standardization, so use standardized protocols and maintain detailed records of all experimental parameters, including temperature, pH, exposure time, and chemical concentration for reproducibility. So when we do these kind of assays, we need to record each and every parameter because these are very simple methods. Okay, so to maintain the reproducibility, it is very, very important to record each and everything. Then the replication, it should perform at least 3 independent experiments with multiple bulbs per treatment group to ensure the statistical validity and account for biological variation. Then, as in all other methods, in here also we need to use the negative control and the positive control. So the negative control is the distilled water, and the positive control may be any substance that can cause the genotoxicity or the cytotoxicity. In other than to that, sometimes when we work with the plant extract material, we need to use some other solvent also to dissolve that plant extract. So if we use that kind of any solvent, then we need to do a separate control for that particular vehicle also. Then the blind analysis, when possible, perform microscopic scoring in the blinded manner, where the observer is unaware of treatment groups to eliminate unconscious bio aberrations identification. This is also important. So, if it's a group studies, normally we are asking one student to prepare the sample and another student to count it. Then the counting student is blind about the sample. So, she or he doesn't know whether which sample is it. And when come to the advantages of this method, it's a very cost effective and accessible, because we need very minimum requirement in at the beginning. Also, I mentioned globally, although there are a lot of alternative methods are available, depend on our funds, depend on our lab facilities. We cannot apply each and every method to our laboratory. So always through our societies, we are trying to introduce these simple methods to our students, especially for the undergraduate students for their primary screening processes. So we need very minimal requirement like the onion bulb and the basic glassware and the microscope. And also there are operational advantages like the low material cost per sample, and we need minimal space requirement, and also simplified training procedures. Then the cytogenetic clarity. This assay provides exceptional visualization of chromosomal alteration, making it ideal for detecting and characterizing genotoxic effects. So the root meristematic cells, they have high mitotic activity, enables observations of chromosomal abnormalities, including breaks, fragments, and irregular division. Then the direct visualization. The microscopic reveals the structural damage with clarity, eliminating in interpretation. Then countable endpoints like the measurable parameters like mitotic index, abbreviations frequency, and the cell cycle phases provide robust data. And also high sensitivity and detecting low level exposure. The Allium-SEPA assay demonstrates exceptional sensitivity for identify even trace concentrations of genotoxic substances, providing early warning signals for environmental and the occupational hazards. And these are the things that we need to consider: the detection time and the response time and the accuracy rate. And also when come to the environmental responsibility, there is a zero hazard waste, the biodegradable materials, and the minimal chemical requirement eliminate the complex waste management challenges and environmental contamination risk. And also in here there is ecological integrity and no animal welfare concern in this method, and no bioaccumulation potential, and complete alignment with green chemistry principles and sustainability goals. And when compared to the traditional animal models, we know that when we get the traditional animal testing for the toxicity studies, it's a high cost and maintenance requirements are needed. Then especially there are a lot of ethical concerns and regulatory restrictions, and also the lengthy experimental timelines and the complex data interpretation. But in this Allium-SEPA assay, we need very minimal resources, and also ethically sound approach, and the rapid results, and straightforward and the reproducible data. And in Sri Lanka, we are using this method for most of our undergraduate research project, and one group worked on what is commercially available plastic water bottles, and they store those water bottles in different conditions, because most of the time that we can see that the people are put their plastic water bottles in the vehicles, and they park their vehicles without any shade, and directly directed to the sunlight, and they keep that bottle 1-2 days within their cars, and they are consuming that. So we store these water bottles in different conditions. Like one group is at the room temperature, another group was at the refrigerator, then another test group was at the cupboard, and another one is at the car that park without any shade. Then we check those water quality, the water bottles by using this method, and at there we saw that there are compared to the negative control, there are some toxicity effects in the bottles which store in the car, the exposure to sunlight, and also in the refrigerator. However, this is an undergraduate project, and they worked on only one brand of commercially available water bottles in Sri Lanka, and we are trying to expand this study for the different brands, and also with the increase of the sample size to get the very clear idea about this, and also needed to check these water for the bisphenol and the phthalate content. So those research are ongoing, and this is one of the publication of abstract publication that we use this method to check the water quality of plastic water bottles. And as I mentioned, throughout my research, when we do the animal studies, always we need to adhere this 3R concept: the replacement, reduction, and the refinement. And under the replacement, we should go to the alternatives. But when we go to the alternatives, always we should select the alternatives which give the scientific validity as well as to the reachable one. Although there are very advanced methods from the undergraduate level, we don't have funds to do that. That is why we are awareing our students regarding this simple but reproducible method. So when come to this Allium-SEPA model, it supports the global ethical standard, the replacement because no need of animals, and the reduction, the health screen chemicals before using more complex tests, and the refinement in here, there are no any animal suffering because we are not using animals. And when come to the key factors, this is simple yet powerful. The Allium-SEPA assay provides the robust cytotoxic and the genotoxic and mutagenic data without complexity. And this is good for the sustainable science because it offers an ethical, cost-effective, and environmentally responsible alternative to animal testing. And this is ready for implementation in any laboratory with very low facility. So anyone can practice this method. So with that, I would like to wind up my talk. So finally, I would like to say that in every dividing cell of an onion roots lie a powerful message: innovation can be human. Okay. So thank you very much for listening, and again thank you very much for the Dr. Sangeeta and the organizing team to inviting me. It is my great honor and the pleasure to share some of our experience with you. Thank you.
Ma'am, are these models mandatory in
Sorry.
are these models mandatory in animal drug testing, like
This
veterinary medicine or something?
this method.
Yeah.
This method we can use only for the genotoxicity and the cytotoxicity testing.
Okay.
So in the drug development also, for the as a primary screening, I think that if you are working with the natural product and you need to check your plant extract has a toxicity, so that kind of primary screening you can use this method. But in the normal development methods, there is a point that you must use animals also.
Exactly. That would be after.
Yeah.
Before that,
So the primary screening you can use for the cytotoxicity and the genotoxicity, but not for the all toxicity style, because in my slide I show there are different type of toxicity.
yeah.
So this is only for the cytotoxicity and the genotoxicity. Especially we can use this for the checking the water quality, that kind of thing.
So is this acceptable globally?
Yes.
Okay.
Is under OECD guidelines?
This one I'm not sure, but there is another method that's called the duckweed under the OECD. So we are practicing the duckweed toxicity study also for to test it. Yes, the cost effective. That is why I said yes. Although there are very advanced methods globally, if we don't have fund and if we don't have facilities, then we should find some simple methods which can give the reproducible results.
This is a very good model for students
Yes.
or any budding research.
Especially for the undergraduate
Yeah.
students, they are recommending this method in Sri Lanka.
Yes.
Dr. Rose, you want to add to it? So can it be used as a non?
Well, thank you, Dr. Kalpani, for this presentation. I just want to know, like, have you validated this model?
We didn't validate, but as I showed in during my presentation, always we are trying to maintain all parameters and get the replicates. Likewise, we are following the protocol, but we didn't validate.
So, are you for future step? Are you interested in means interacting with regulator or?
Yes, I also hope so that if we can validate this method, it's better. So in the future, we should try for that.
Thank you.
Yes, it's good. But I think even like sir said, you should have some comparative studies during the validation, so you can prove that and get in touch with the regulators.
Yes,
And
so
start with some comparative studies.
we'll try out. Yes, in Sri Lanka, we have only very few cell culture labs also. Yes.
Yes. Okay, thank you, Dr. Kalki, for insightful presentation on William Sipa as a non-animal monitor in the toxin cell. And it's a powerful cost-effective medical cell therapy. Then by those researchers, we cannot go for cell and cell. Thank you so much. Now, I request Dr. Chandra Rao to share something. But as Dr. Ujjwal has very beautifully covered every and each aspect of organoid, so I think we should stop here. And Dr. Rodit wants to interact with the students, so he can interact till the lunch with them. And if you have any queries regarding organoids, you can discuss.